We are analyzing Chapter 2, Water and pH, from a pretty rigorous biochemistry text.
Oh, yeah.
We are going to deconstruct the chemical physics of this molecule line by line.
And when you do that, when you really strip away that familiarity water, turns out to be, well, a bizarre molecule.
Yeah.
It's chemically aggressive, mathematically precise.
Yeah.
It's a matrix.
Not just a filler.
Not at all. It dictates the shape of every single protein in your body. And it literally uses a logarithmic scale to decide if you live or die.
We're going to cover everything from the geometry of the electron orbitals all the way to the derivation of the Henderson-Hasselbalch equation. But first, let's establish the stakes. The chapter opens with a section on biomedical importance. So why does a clinician really need to understand molecular geometry?
Because the body is, in essence, a bag of water that is constantly regulating itself against chaos. That's really what it is.
It's a bag of water.
Water solvates everything, all the organic and inorganic molecules, which allows them to move and interact. But it's also an attacker. It's an excellent nucleophile, which I'm sure we'll get to.
Oh, we will.
The text highlights that the regulation of water is just critical, and it involves these complex hypothalamic mechanisms, the antidiuretic hormone, or ADH.
And the kidneys, of course.
And the kidneys. And when that regulation breaks, it is absolutely catastrophic.
The text details a specific condition here that really stood out to me. Nephrogenic diabetes insipidus.
Yes, this is a perfect illustration of the whole system failing. So normally, ADH tells your kidneys to conserve water.
To make your urine more concentrated so you don't dehydrate.
Exactly. But in this condition, the kidneys just stop listening. The receptors fail. They go deaf to the signal.
So it doesn't matter how much water you drink.
Nope.
You can't retain it.
You can't. You lose the ability to concentrate urine, which then triggers this unquenchable thirst mechanism. It's a total systemic failure. And it's all caused by a breakdown in water management.
But even more precise than the volume is the regulation of acidity, right?
Oh, much more. The pH balance, the window here is just incredibly narrow.
How narrow are we talking?
Your extracellular fluid, so your blood, basically, it has to be maintained between a pH of 7.35 and 7.45.
That's it. That's the entire margin of error.
If you drop below 7.35, you enter a state of acidosis.
Which can be caused by things like diabetic ketosis or lactic acidosis from extreme exercise.
Right. And on the other side, if you go above 7.45, you hit alkalosis. This can happen from something as, seemingly simple as severe vomiting, losing all that acidic content from your stomach.
So the point is, your enzymes, your oxygen transport, your entire cellular structure, they all rely on this razor-thin mathematical balance.
They do. And to understand how the body holds that line, we have to start with the molecule itself.
Okay, let's zoom in. Section 1. The geometry of the water molecule. We all know the cartoon version, the Mickey Mouse ears, 1 oxygen, 2 hydrogens. But the text gets really, really specific about the angle.
You have to visualize this in 3D. It's not flat at all. It's a tetrahedron. Or, more accurately, an irregular, slightly skewed tetrahedron.
Okay, let's unpack that skewed part. Why isn't it a perfect pyramid?
It all comes down to the oxygen atom at the center. Oxygen has what we call sp3 hybridized orbitals. So you can imagine that pyramid shape. 2 corners of that pyramid are occupied by the hydrogen atoms.
And the other 2?
The other 2 corners are occupied by unshared electron pairs.
So these are electrons that aren't actually bonding with anything. They're just sitting there on the oxygen.
They're just hanging out. And chemically, they are large, they're diffuse, they take up a lot of space. In a perfect tetrahedron, like, say, a methane molecule, the angle between the bonds is a perfect 109.5 degrees.
But not in water.
No. In water, those big unshared electron pairs repel each other. They're both negatively charged, so they push against the structure.
Squeezing the hydrogen atoms closer together.
Exactly. They literally crunch that angle down from 109.5 to just 105 degrees.
And does that 4-degree squeeze really matter that much?
It matters immensely. It creates the specific bent shape that drives the molecule's polarity. And that right there leads us to the most important concept in this entire section: electronegativity.
Oxygen is an electron hog.
It's a bully. It is strongly electronegative. It yanks the shared electrons in the covalent bond away from the hydrogen nuclei.
So they spend more time around the oxygen.
Way more time. So the oxygen side of the molecule becomes partially negative, and the hydrogen side, which has been sort of stripped of its electron density, becomes partially positive.
And this creates a dipole. Two poles, a positive and a negative end.
And that dipole is what makes water the universal solvent.
Yeah.
This is where the text introduces a concept that I think often confuses people, the dielectric constant.
Right. It gets a little math heavy here with Coulomb's law. It says F, the force of interaction between two charges, is inversely proportional to this constant epsilon.
A simple way to think of the dielectric constant is insulation, or maybe like magnetic shielding.
Okay.
Coulomb's law just asks, how strongly do two charged particles attract each other? You have a positive ion and a negative ion. They want to snap together. The dielectric constant is the ability of the medium they're in to get in the middle and stop that from happening.
So it weakens the attraction.
It weakens it. Let's
Yeah.
look at the data points in the text because they tell the whole story.
Yeah.
In a vacuum, epsilon is 1. There's no shielding at all.
Full strength attraction.
Exactly. Now, in hexane, which is a nonpolar, oily kind of solvent epsilon, is about 1.9.
Still very low.
Very low. So if you put table salt sodium ions and chloride ions into hexane, they will find each other almost immediately. The attraction is just too strong. The hexane can't stop them, so the salt won't dissolve.
But then you look at water. At 25 degrees Celsius, the dielectric constant is 78.5.
That is a massive, massive jump from less than 2 to almost 80.
So what is physically happening to the salt ions in the water?
The water molecules swarm them. Because water is a dipole, the negative oxygen end of the water molecules orient themselves around the positive sodium ion.
And the positive hydrogen ends surround the negative chloride.
Precisely. And because that dielectric constant is so high, the water effectively screens or insulates those charges from one another. It reduces the force of attraction between them so significantly that they just drift apart.
And that is the very definition of dissolving. It's not some kind of magic, it's just electrostatic insulation.
That's all it is.
Okay, so that's how water interacts with ions. But section 2 is about how water interacts with itself. We're talking about hydrogen bonding. The text calls this the glue of life.
And it really is. This is the mechanism that keeps water a liquid at room temperature. If you just look at the periodic table, a molecule this light should be a gas. It should be a vapor.
Like hydrogen sulfide.
Exactly. But hydrogen bonds hold it all together.
So how does the bond actually form? I mean, it's not a covalent bond. It's not actually sharing electrons.
No, it's purely electrostatic, but it's a very specific kind of electrostatic interaction. You need a hydrogen atom that is covalently bonded to an electron withdrawing atom, like an oxygen or a nitrogen.
That's what leaves the hydrogen naked, right? Partially
Yeah.
positive and unshielded.
That's the key. And so that naked proton is looking for some stability.
And it finds it. Where?
It finds it in an unshared electron pair on a neighboring atom. In water, the hydrogen of molecule A is attracted to one of the lone pairs of electrons on the oxygen of molecule B.
And since every water molecule has 2 hydrogens to donate and 2 lone pairs to accept.
It can play both sides. It's both a hydrogen bond donor and an acceptor. And this allows water to form these massive interconnected networks. The text mentions ordered arrays or clusters.
But here's the paradox that really stood out to me. The text talks about this grand structure, but then it immediately emphasizes how weak and fleeting these bonds are.
That's the critical nuance. That's the whole magic of it. The bond energy is only about 4.5 kilocoulomb.
How does that compare to a real bond?
Well, the internal OH covalent bond inside the water molecule is about 110 kilocoulomb.
So a hydrogen bond is what, less than 5% of the strength?
It's a whisper of a bond.
Yeah.
And the timing is even more dramatic.
Go on.
The half-life of a hydrogen bond in liquid water is a few picoseconds.
A picosecond, that's a trillionth of a second?
Yeah, so you shouldn't visualize a static scaffolding, like a crystal lattice. You should visualize a seething, flickering mass. Bonds are breaking and reforming billions of times per second.
So it's not a solid structure, but a statistical one.
That's the perfect way to describe it. Statistically, at any given instant, each water molecule is bonded to an average of about 3.5 others. It's structure in motion.
And this isn't just limited to water-water interaction.
No, not at all. The text highlights figure 2.3, which shows hydrogen bonds are everywhere in biochemistry. Alcohol molecules do it. Peptide bonds in proteins do it. The carbonyl oxygen of one amino acid bonding to the nitrogen's hydrogen of another.
Which is the fundamental basis of protein folding, the alpha helix and the beta sheet.
It's the zipper. It's the molecular zipper that holds the entire shape of the protein together.
That transitions us perfectly into section 3, forces that stabilize biomolecules. So we have the covalent bonds, which are like the steel beams holding the atoms together.
They're the primary structure.
But the text focuses on the non-covalent forces as the things that determine the final functional structure.
Right. If covalent bonds are the anatomy of a molecule, The non-covalent forces are its physiology. They allow for movement, for interaction, for function.
And the most misunderstood one has to be the hydrophobic interaction. We've all heard oil and water don't mix. And the common assumption is that oil molecules really like each other.
Like attracts like, right. That's what we're all taught.
Exactly. But the text explicitly, and I mean explicitly, corrects this. It says, quote, "It is not driven by mutual attraction between the nonpolar molecules. It is driven by entropy."
This is a concept that breaks a lot of people's intuition. It's one of the hardest things to grasp in introductory biochem. You have to stop looking at the oil and start looking at the water.
Okay, walk us through the mechanism. What happens when I drop a single nonpolar molecule, like an oil droplet, into a beaker of water?
The water molecules right at the surface of that oil droplet can't hydrogen bond with the oil. Looks like they've hit a wall.
A nonpolar wall.
Right. But water molecules are obsessed with maintaining their hydrogen bonding network. So to compensate, the water molecules that surround that oil droplet form a highly ordered, rigid cage.
The text calls it a clathrate cage, I think.
Yes, that's the technical term. The water molecules essentially freeze into a specific orientation to maximize their bonds with other water molecules while still avoiding the oil.
But being frozen in a specific orientation, that's a state of high order.
And in thermodynamics, order is expensive. Order means low entropy, low randomness. And the second law of thermodynamics says that the universe always wants to move towards chaos, towards maximum entropy. So having all this water trapped in these ordered cages is energetically unfavorable.
So how does clumping the oil together fix this problem?
It's all about geometry. Imagine you have two separate small oil droplets. You have two separate water cages around them. That's a lot of trapped, ordered water.
Right.
But if two droplets merge into one big blob,
The total surface area decreases.
exactly, you know your geometry. Less surface area means a smaller cage is required. Fewer water molecules are needed to surround the single big blob.
So the extra water molecules that were part of the two smaller cages are released.
They're set free. They go back into the bulk liquid.
They regain their freedom to tumble and rotate.
Their entropy skyrockets. And that increase in the entropy of the water is the thermodynamic driving force. The universe isn't pulling the oil together. It's pushing the oil together just to liberate the water.
That really flips the script. The hydrophobic effect is basically just water demanding its personal space back.
That's a fantastic way to put it. And this force, this entropy-driven force, is what drives proteins to fold their hydrophobic cores inward, and it's what drives the spontaneous formation of cell membranes.
Amazing. Okay, let's briefly touch on the other two forces mentioned in this section: electrostatic interactions and van der Waals forces.
Electrostatic interactions are much more intuitive. They're often called salt bridges. It's just the attraction between opposite charges.
Yeah.
A positively charged lysine side chain attracted to a negatively charged glutamate side chain.
And they're strong.
They're comparable in strength to hydrogen bonds, but they have the advantage of being able to work over longer distances.
And then there's van der Waals. The text calls them transient dipoles. That sounds weak.
They are the weakest of all, individually. But their power comes from numbers. They are everywhere.
How do they form?
Even in a perfectly neutral atom, the electrons are constantly zooming around the nucleus. So for a split femtosecond, you might have more electrons on the left side of the atom than the right.
And that creates a tiny temporary dipole.
A fleeting one. But that tiny dipole can then induce a complementary dipole in a neighboring atom. It's like a very, very faint flickering magnetism.
And they are incredibly distance dependent.
Insanely so. The text notes the force decreases as the sixth power of the distance. That's 1 over r to the sixth.
So if you just double the distance, the force drops by a factor of 64.
It vanishes. It drops to 0 almost instantly. But if you push them too close together, their electron clouds clash, and they repel each other strongly. So there is this perfect Goldilocks distance.
The van der Waals contact distance.
Right. It's a sweet spot where the faint attraction is maximized right before the repulsion kicks in.
And the text brings all of this together using DNA as the ultimate example.
It's the perfect synthesis. The backbone of each strand is held together by strong covalent bonds.
Unbreakable.
The two strands are held to each other by the hydrogen bonds between the bases, A with T, G with C.
The rungs of the ladder.
Exactly. Then, the bases stack on top of each other like pancakes, and those stacks are stabilized by countless van der Waals forces.
And finally?
Finally, the charged phosphate backbone sits on the outside of the helix, where it can interact electrostatically with water, while the hydrophobic bases are tucked away on the inside. DNA uses the entire toolkit of non-covalent forces.
It's just beautiful. Okay, moving on to section 4. We've been talking about water as a structural element, a solvent, but now we see it as a chemical reactant.
More than that, it's a chemical attacker. And to understand this, we need to define two terms: nucleophiles and electrophiles.
A nucleophile is a nucleus lover.
Which means it loves positive charge because it is itself electron-rich. Think of atoms with lone pairs of electrons, like oxygen, nitrogen, or sulfur.
So water, with its two unshared electron pairs on the oxygen, is a prime nucleophile.
An excellent one.
And the electrophile.
The electron lover. It's electron poor. The classic example is a carbonyl carbon, a carbon double bonded to an oxygen, like in a peptide bond or an ester. The oxygen steals the electron density, leaving the carbon partially positive and hungry for electrons.
So what happens when nucleophilic water meets an electrophilic carbon?
The attack. It's called hydrolysis. The water's lone pair attacks the carbonyl carbon, which eventually leads to the cleavage of the bond. Water splits the molecule and adds itself to the pieces.
And this is how we digest food.
This is how we break down proteins into amino acids and complex carbs into simple sugars. It's fundamental.
But this brings up a massive biological contradiction. The text makes a crucial distinction here between thermodynamics and kinetics.
Yes, this is the difference between what should happen and what will happen. Thermodynamically, hydrolysis is favorable. It releases energy. It's a downhill process.
So if you look at the energy balance, the proteins in my body should spontaneously break down into a puddle of amino acid soup.
They should. We are all thermodynamically unstable.
That's a comforting thought.
It is, isn't it? But kinetically, the reaction is incredibly slow. There's a high energy barrier, what we call the activation energy, that prevents the water from just randomly attacking all our peptide bonds.
That barrier is the only reason we don't dissolve.
Pretty much. And this is where enzymes come in. Enzymes like proteases or nucleases act as catalysts. They grab the reactants and hold them in just the right way to lower that activation energy barrier.
So they don't change the should, they just change the will. They make it happen now.
They speed up the reaction rate by orders and orders of magnitude, but only when and where the cell wants it to happen. They give the cell kinetic control over the thermodynamic inevitability.
Okay, that's a perfect pivot. We're now going to shift to the second half of the title, pH, Section 5. The math of dissociation. This all starts with the simple fact that water, on occasion, just falls apart.
It does. It's the ionization of water. A water molecule has a very slight tendency to dissociate into a proton, which we write as H plus an, and a hydroxide ion, OH.
And the text clarifies that free protons, H plus E, don't actually exist in water.
No, a naked proton is far too reactive. It would be an insane chemical entity. It immediately gets hydrated by another water molecule to form a hydronium ion, H3O plus 8.
But we just write H plus for simplicity.
By convention, yes. Yeah. Biochemists are lazy. We just write H plus to keep the math clean.
Speaking of the math, the text gives us a probability. How often does this dissociation actually happen?
It's incredibly rare. The probability of any given hydrogen atom in a sample of water existing as a free ion is about 1.8 times 10 to the minus 9.
That's 1 in about 500 million.
Roughly, yeah. For every one hydrogen ion, there are about 550 million intact water molecules.
So if it's that rare, why on earth does it matter?
It matters because of the sheer density of water. We need to do a classic calculation here. What is the molarity of pure water? What is the concentration of water in water?
It sounds like a riddle.
It's just simple mass over volume. 1 mole of water is 18 grams. You've got H2O, so 16 for oxygen, plus 1 for each hydrogen.
All right, 18 grams per mole.
And 1 liter of water is 1,000 grams. So you just divide 1,000 grams by 18 grams per mole, and you get
55.56 molar.
55.56 molar. Just stop and think about that number for a second. When you do chemistry in a lab, a really strong concentrated solution might be 1 molar, maybe 5 molar. Water is 55.56 molar.
It's incredibly concentrated in itself.
It is. So even if the probability of splitting is 1 in half a billion, when you multiply that tiny probability by the enormous concentration, 55.56 molar, you end up with a significant absolute number of ions.
And this leads directly to the derivation of K-DOA, the ion product of water.
Exactly. The full equilibrium constant would have the concentration of water in the denominator. But since that concentration, 55.56 Pa, is essentially constant, we just bundle it into the constant itself.
And we're left with a simpler expression.
We're left with Kw equals the concentration of H plus times the concentration of OH.
And at 25 degrees Celsius, this product always equals
10 to the minus 14, that is the magic number for all of aqueous chemistry. It's a constant. It means the product of protons and hydroxide is always 1014.
So in perfectly pure neutral water, they have to be equal.
Which means the concentration of H plus is 10 to 7, and the concentration of OH is also 10 to 7.
And 10 to the minus 7 is a really annoying number to write down all the time.
It is. So in 1909, a chemist named Sorensen gave us a gift. He gave us the pH scale, section 6.
The P function.
The P function simply means take the negative logarithm of, so pH is just shorthand for dash log H plus guy.
Let's quickly clarify the implications of using a negative log scale. There are two big ones.
First, it inverts the scale. A high concentration of protons gives you a low pH, so acids have low pH.
And second, it's exponential. It's a logarithmic scale. A change of just one pH unit is actually a 10-fold change in the real concentration.
That's so important to remember. So pH 7 is neutral. pH 6 has 10 times more acid than pH 7.
And pH 5 has 100 times more acid.
Correct. It's a logarithmic ladder.
Let's run through the trick calculation in the text. It asks, what is the pH of a solution with a hydroxide concentration, an OH, of 4.0 by 10 to 4 molar?
The trick, of course, is that you can't just plug that number into the pH equation, because it's hydroxide, not hydrogen ions.
So you have to go through the back door.
You have to find the pOH first. So you take the negative log of 4.0 by 10 to 4.
Which comes out to 3.4.
A pOH of 3.4. And since we know that pH plus pOH must always equal 14,
you just subtract. 14 minus 3.4 gives you a pH of 10.6.
which tells us immediately that it's a basic or alkaline solution.
Right. Okay, so water is the baseline, but it gets interesting when you start adding other things to it. Section 7, acids, bases, and pKa.
And first we need to distinguish between strong and weak acids.
Strong acids, like hydrochloric acid, HCl.
They dissociate completely. If you put them in water, they dump all of their protons. It's a one-way street.
But biology doesn't really run on strong acids.
Not at all. Biology runs on weak acids. Things like amino acids, DNA bases, various metabolites. They don't just dump all their protons. They exist in an equilibrium. They hold on to some protons and they release others.
There's a tug of war.
A constant tug of war. And to quantify that stickiness for the proton, we use the acid dissociation constant, Ka.
Which, like the hydrogen ion concentration, is often a small and clumsy number.
Exactly. So we use the P function again. We convert that Ka into a clean, simple pK.
pK equals dash log Ka. What's the simple rule for interpreting pK?
It's an inverse relationship. The stronger the acid, the lower its pK. A low pK means it gives up its proton very easily. A high pK means it holds on tight.
The text gives a few examples. Acetic acid, which is vinegar, has a pK of 4.76. The ammonium ion NH4 plus A has a pK of 9.25.
So between those two, acetic acid is by far the stronger acid. It's much more willing to donate its proton.
What about something like phosphoric acid? The text calls it polyprotic.
Polyprotic just means it has more than one proton to give away. Phosphoric acid, H3PO4, has 3.
So does it have one pK?
No, it has 3 distinct pK values, one for each proton. 2.15, 6.82, and 12.38.
And why do the numbers jump up so much? Why is the third proton so much harder to remove than the first?
It's all about electrostatics. When it loses that first proton, the molecule becomes negatively charged. It becomes H2PO4.
And that negative charge is going to attract the remaining positive protons.
It holds onto them much more tightly, so it becomes harder to pull the second one off, and the pKa goes up. Then after the second one leaves, you have a -2 charge, HPO4, which holds on to that last proton for dear life. So the pKa skyrockets.
That makes perfect sense. Okay, we have arrived at section 8, the part of the chapter that gives students nightmares. The Henderson-Hasselbalch equation.
It really shouldn't be scary. In fact, it's the opposite. It's the Rosetta Stone of acid-base chemistry. It's what lets you connect pH, pKa, and the ratio of the acid and its conjugate base.
Hey, let's derive it. I think walking through it makes it less intimidating. A verbal math time.
Okay. Step 1. Start with the equilibrium expression, Ka equals H plus AHA. That's protons times the conjugate base divided by the acid.
Step 2. We want to solve for H plus H.
Right. So we just rearrange the terms. H plus Ka times the fraction HAA.
Step 3. Take the logarithm of both sides.
So you get log H plus ACO log Ka plus log HA.
Step 4. Now we convert to P notation by multiplying the whole thing by -1.
And S log H plus becomes pH, das log A becomes pK. So the equation is now pH equals pKa log HA.
We're almost there. Step 5. I don't like that minus sign. How do we get rid of it?
We use a basic log rule. Dash log xy is the same as plus log yx. So we just flip the fraction.
Yeah.
The base A goes on top, and the acid HA goes on the bottom.
And the final equation is?
pH pK plus log AHA.
Okay, so why is this equation so famous? What's the aha moment it provides?
The aha moment comes when you look at the special case, the half neutralization point. Ask yourself, what happens when the concentration of the base A is exactly equal to the concentration of the acid, HA?
Then the fraction AHA is just 1.
And what is the log of 1?
0.
0. So that whole log term just disappears, and the equation collapses to its most beautiful, simple form, pH equals pKa.
This is the anchor point. If you know the pK of a molecule, you know the exact pH where that molecule is 50% protonated and 50% deprotonated.
And that concept, right there, is the entire foundation of Section 9. Buffers and titration curves.
Let's try to visualize the titration curve shown in Figure 2.5.
Okay, so imagine you have a beaker full of a weak acid, like acetic acid. You start slowly dripping in a strong base, like NaOH, and you measure the pH as you go.
The pH starts low and begins to rise, but it's not a straight line.
No, it traces a sigmoid shape, an S-curve. And the most important part of that S-curve is the middle, where it's flattest.
That flat region is the buffer zone.
That is the buffer zone. In that region, you are adding a strong base, which should be making the pH skyrocket, but the pH is barely changing. It's resisting the change.
Why? What's happening chemically?
It's because you have this big reservoir of the undissociated weak acid, the HA form, and it's stepping up to the plate. As the incoming OH tries to raise the pH, the HA donates a proton to neutralize it, turning it into water.
It's soaking up the punch. It's sacrificing itself to keep the pH stable.
Exactly. And that buffering capacity is at its absolute maximum right at the midpoint, where pH equals pKa, because you have equal amounts of the proton donor and the proton acceptor.
And the effective range for a buffer is usually considered to be what?
The rule of thumb is the pKa plus or minus 1 pH unit.
Yeah.
So for acetic acid with pKa of 4.76, it's a good buffer between about 3.76 and 5.76.
If you push beyond that range,
You exhaust the reservoir. The ratio of base to acid becomes too extreme, like 100 to 1, and the buffer breaks. The pH will then spike or plummet.
and our bodies rely on this constantly.
Every second of every day, normal metabolism produces a huge amount of acid. Carbon dioxide from respiration, lactic acid from muscle, ketone bodies from fat metabolism. Without buffers, our enzyme systems would crash instantly.
So what are the main physiological buffers that keep us in that 7.35 to 7.45 window?
Bicarbonate is the big one in the blood plasma, regulated by the lungs and kidneys. Phosphate is the major player inside our cells, where its pKa of 6.82 is perfect.
And proteins too, right?
And proteins themselves are significant buffers, especially hemoglobin and red blood cells. The histidine amino acid side chains have pK values right around neutral, so they are excellent at soaking up and releasing protons.
This brings us to the final section, Section 10, the effects of the molecular environment. We tend to think of a pK as this fixed, constant number you just look up in a table, but the text says, not so fast.
Not at all. The local environment can change a pK dramatically. The pK of a molecule depends entirely on the medium it's in.
And we're back to the dielectric effect again.
We are. So take a carboxylic acid. In water, with its high dielectric constant, it's happy to dissociate and form a negative charge because the water can solvate and stabilize that charge.
But what if you take that same carboxylic acid group and bury it deep inside the oily hydrophobic core of a protein?
Now you're in a low dielectric environment. There's no water to shield that charge. It becomes energetically very, very difficult to create that charge in the first place.
So the acid refuses to give up its proton.
It holds on tight, its pKa shoots up, it becomes a much weaker acid than it would be in pure water.
And what about the proximity effect?
This is all about electrostatic crowding. The text gives a great example comparing succinic acid and glutaric acid.
They both have 2 carboxyl groups.
They do. But in succinic acid, those 2 groups are very close together, separated by just 2 methylene groups. In glutaric acid, they're further apart, separated by 3.
Okay, so how does that change things?
Let's look at succinic acid. When the first carboxyl group ionizes and becomes negatively charged, that negative charge is sitting physically right next to the second group.
And negative repels negative.
Right. So that first negative charge actively pushes against the second group, releasing its proton. It makes it harder to remove the second proton. It raises the second pK significantly. In glutaric acid, the groups are further apart, so the interference is much less.
It's just amazing how pure geometry dictates chemical reactivity.
Structure determines function. That is the central dogma of all of biochemistry, and it starts right here with the simple geometry of water and the location of protons.
Okay, let's try and bring all of this together. We started this whole deep dive with a simple glass of water.
And we saw that it's not simple at all. It's a collection of bent, polarized, tetrahedral molecules.
That form these flickering hydrogen-bonded clusters that only last for picoseconds at a time.
That use the laws of entropy to force nonpolar molecules to assemble into membranes and protein cores.
And that use a delicate logarithmic mathematical probability to maintain a precise pH that literally keeps us alive from one moment to the next.
It's a matrix. It's not just a passive solvent. It's the stage, the scenery, and the director of the entire play of life.
The text mentions, and I love this phrase, that life exists on the edge of chaos, balanced by these incredibly weak reversible forces.
That is the ultimate takeaway. We aren't held together by unbreakable stone walls. We are held together by handshakes, by fleeting hydrogen bonds, by faint van der Waals forces, by these delicate buffer equilibriums. It's all dynamic. It's all in motion.
So next time you drink that glass of water, maybe take a second to remember the sheer chaos and the beautiful math that's swirling around in there.
It's quite a cocktail, a seething mass of flickering clusters and proton hopping that makes all of this possible.
Thanks for diving deep with me today.
My pleasure.
We'll see you in the next one.